Hello, students and staff and faculty here at NIH. I would like to thank you, and NINDS. I would like to thank you for the invitation to speak here. I am truly honored, and I'd like to thank Michelle for that generous introduction. She stuck with a lot of the good stuff and avoided maybe some of the bad stuff. I don't know if I'll avoid the bad stuff during my presentation. Um, But I noticed that the presentation I started with had my previous and somewhat current affiliation at the Neuroscience Institute at Morehouse School of Medicine, where I've been for the last 10 years. But as Michelle has noted, I've had to update that because as of last week, I now am an associate professor at UCLA in the Department of Integrative Biology and Physiology. And I'm going to talk to you in talking about my journey today. I'm going to give you a few details about how that happened. So my presentation is going to be in three parts, so I had planned it to be in three parts. The first 2 or 3 slides, I'm going to talk about some of the recent science that we've published in my lab, and then a few slides after that, I'm going to be going through my journey, actually from grade school to where I am now. And then after that, I have a series of slides where I'm going to talk about what it takes to be successful as a student at every level. However, as I walked in and saw the students filing in, and as I sat down and took a look out at the audience, I decided just a few minutes ago that I would spend a little bit more time on my journey because I think that there are some details about my personal experience that will hopefully resonate with some, if not most, of the students here. And I think that some of the details of my personal experience are also very relevant to a lot of the social issues that are going on today, and that some of those issues are important for young scientists, young doctors, and young engineers. So bear with me as I go through my journey. I told Michelle at the beginning I was not going to talk about some of these things, but as I see such bright young people out here, I feel like I should take the opportunity. All right, so what do? I study sleep, and right here are a list of the 2 primary processes, the targets that my lab focuses on. Our goal is really threefold. The first is to try to treat and cure sleep disorders. Sleep disorders many of you are familiar with, insomnia, narcolepsy, some sleep disorders you may not be as familiar with. We're trying to find more effective targets of treating sleep disorders. The second is to help people who are having problems sleeping just sleep better. You may have issues sleeping because of a variety of reasons, because of the environment, because of family obligations, because you're in school and you have a test tomorrow and you're very anxious or you have not studied, so you have to stay up studying. I know there's probably 1 or 2 people in the audience that can relate to that. Another of our goals are just helping people sleep better in our increasingly 24-hour society. And the third goal is to try to help people that are not able to sleep. Many people have situations that prevent them from sleeping. If you're in the military in an occupied or a war zone and you have to fight, a lot of times sleep opportunities are few and far between. If you're a resident on call, sometimes sleep opportunities are few and far between, and even when those opportunities present themselves, you might actually try to use that to work to get ahead instead of sleeping. Or if you are working in a disaster area, a catastrophe because of a natural disaster, and you're on an emergency response team, a lot of times you don't have as many sleep opportunities. But we know that lack of sleep clouds your mind, it inhibits your cognitive capabilities, and in the long run, it hurts your health. It increases the risk for a variety of diseases, chronic and infectious diseases. As a field, we've been telling you the same thing since I've been in the field, and probably a lot longer than that. You need to get, how many hours of sleep? Who can say, how many hours of sleep a night do you need to get? 8 hours, between 7 and 8.5 hours a night. And we've been saying that for years, and every year it seems we've been having less and less success. The drive to stay awake seems to be increasing, and our ability to resonate is getting smaller. So we have an additional responsibility, and that is to try to help people who are not sleeping find a way to preserve their cognitive abilities. If you're working on an emergency response team, an error caused by sleep deprivation could be critical. So it's responsible for us to try to find ways to help people who aren't able to sleep preserve their psychomotor vigilance, their cognitive abilities, and more importantly, their health in the long run. The primary systems in my lab that we target are the circadian timing system, which as Michelle noted, I studied in graduate school, and sleep homeostasis. We know a lot about circadian rhythms. We don't know very much about sleep homeostasis, and this is the primary focus of my lab. All of the projects in my lab seem to find synergy in looking at sleep homeostasis, which most easily defined is the ability to recover from sleep loss. So when you've lost sleep, when you've been studying for a test or you've been preparing, to do something in medical school, after you've done that, the next time you have a sleep opportunity, you should sleep longer and you should sleep more deeply. That's a biological mechanism that allows you to recover from the lost sleep. And more importantly, that mechanism allows you to recover the cognitive abilities that you sacrificed when you lost sleep, your psychomotor vigilance, and in the long term helps you preserve your health, help you fight off infectious diseases, and things like that. Well, we don't know very much about sleep homeostasis. So my lab is really focused on finding genetic and molecular targets to which we can act on this mechanism for people who aren't able to get sleep and help them preserve the things that make life more difficult. So the goal for our lab is to identify the genes and molecules that regulate sleep homeostasis. And we have several experiments and several projects right now that are moving toward that goal. The majority of them funded by NINDS. So again, it's a great honor for me to be here and talk about what I'm doing to the people that are helping me do it. And actually, it's a good skill you guys should learn early on to be able to talk about what you do, not only to a broad audience, but particularly to the people that are helping you do it. This is a paper we recently published in Journal of Neuroscience that reports one of the results of those projects. This is a gene UB3A whose mutation disrupts sleep homeostasis. As I said earlier, sleep homeostasis is the ability to recover from sleep loss, but it leaves circadian rhythmicity largely intact. And that's very important because our goal is to be able to act on one mechanism, sleep homeostasis, while preserving the other mechanism. Another unique factor about this gene is that its mutation in humans is only debilitating when it's inherited from the mom. If it's inherited from the dad, it does not cause many problems. And the disease that's associated with is Angelman syndrome. Angelman syndrome is a mental illness that is associated with a large variety of negative sleep and wake phenotypes, which is what we were interested in. So we found that the mutation had, as another group had previously reported, that the mutation had effects on circadian function. Well, we had what we consider a more pure form of that mutation, and we found that it did not have an effect on circadian function, as indicated by the wheel running diagrams here. Here along the x-axis are 24 hours of continuous recording of sleep and wakefulness and of locomotor activity. Locomotor activity is indicated on the y-axis on the left, and relative slow wave activity is reported on the right. Slow wave activity is our measure of sleep depth, or sleep depth when you're asleep, or the best way to describe it is sleepiness when you're awake. Sleepiness is different from slow wave activity. Actually measures what we call your sleep need or your sleep pressure. Sleepiness is one of the outcomes of that sleep pressure. And what we found is in the mice that inherited a deleted gene from the mom, that the circadian rhythm was preserved. Our first initial finding was this siesta that mice have, humans have also, mostly men. This increased sleepiness that occurs during the active phase, in mice is during the dark phase, in humans it's during the light phase, it's during the daytime, which is why as I'm talking to you here about 11 o'clock, most of you are going to be wide awake. If I talk to you about 2:30, half the room would be dozing off. It's this increased propensity to sleep that many of us, but not all of us, experience in the afternoons. And that has a gender difference, which we're also interested in my lab. But this gender difference is the men. The men tend to feel more sleepy in the afternoon than women do, and several studies around the world have reported this. My former colleague Shaki says that's why men are so lazy. I said as long as I have the opportunity to study it, I'll take whatever quips that my colleagues are giving me. So the first one was we found that the mutation eliminated this propensity to sleep. We also found that the mutation decreased sleep pressure. It impaired the wake-induced sleep pressure, which normally rises when you're awake. When you're awake, as you can see, your propensity to sleep increases, and we found that that was inhibited by this mutation. It caused the elimination of this siesta and selectively disrupts process H, which is sleep homeostasis. So this, again, we just recently reported on this. We think is a pretty important finding, and we recently obtained funding from NINDS to pursue this finding with a 2-year award on our U54 grant. So this is just a snapshot of the things that we're doing in my lab. We're using kind of hard-wired biological systems to try to understand how our sleep-wake cycle works, particularly in stressful situations, and trying to find targets to better treat sleep-wake disorders. This is, again, a slide talking about our findings. So as I said earlier, in humans, UB3A is normally associated with Angelman syndrome. Only the maternal allele is expressed in neurons. The paternal allele is epigenetically silenced in adults. So our mice are heterozygotes, and we studied those with the null allele from the mom. And the mouse models are whole animal null allele, which we used. A Crelox-P knockout, which we're currently funded by NIH to pursue, and we've just begun those experiments, and a Crelox-P rescue, where we're able to selectively knock the gene out and restore it in isolated populations of neurons in the brain. And what we're trying to do right now is identify the specific brain area that this mutation is affecting the sleep-wake cycle. So there's a lot to do. We won't do it, but hopefully throughout my talk, I can convince 1 or 2 of you to become interested in this, and you can come along and pick up the work. So that was a snapshot of some of our more recent results. We're really interested, again, in finding new and novel genes to regulate sleep so we can find more effective molecular targets. But what I'd like to do now is switch gears a little bit and talk about my journey, how I got here, how I got to the point where, coming from a local DC neighborhood where I was interested in science but really did not know how to do it, how to get into it, to where I am now. And so I'm going to tell you a few stories, and some of them may drag on. I was told that I'm good on time. I don't know if that was a good thing to tell me. So I'm going to give you guys a few stories, and hopefully some of them will resonate. I'm not probably going to be able to cover a lot of the advice that I was going to give you, which is why I'm happy that we have a panel later, which will give me the opportunity if questions should arise about that. So I went to Paul Laurence Dunbar Senior High School. It's in downtown DC, not far from Union Station. And this is Paul Laurence Dunbar as it looks today. Anyone who's ridden by First Street near New Jersey Avenue or New York Avenue may have seen it. But this is not the Dunbar I went to. This is a new Dunbar, which I'm very proud of. This is the Dunbar that I went to. I went there in the 80s. I won't say what specific year I went there in the 80s. And I'm going to leave this slide up for a minute because, like I said, as I was sitting, I decided I wanted to talk to you about some of my experiences when I was in grade school. So as Michelle noted, and as if you see the video she cited, I talked about how I wanted to do science since I was a little kid. I can, as early as I can remember wanting to do anything, I wanted to do science. I was always interested in it. I was always intrigued by it. And I'm the only one in my family and the only one out of my close friends who was actually doing what they said they were going to do when they were a little kid. I recently had a lot of family at my house for the 4th of July, and my aunts were just having so much fun with that. He's doing what he said when he's a little kid. Unfortunately, my cousins weren't very happy with that. But anyway, I grew up in the Shaw area of Washington, D.C., which is where my high school was located. And in the late 80s, when I was in high school, D.C. was going through some pretty rough times. We were the murder capital of the country during that time, and I was a teenager living in one of the more violent neighborhoods in the city. So I lost a lot of close friends to that violence. Dunbar wasn't doing as well as it is doing now either. So oftentimes just getting to school in the morning would be somewhat of an ordeal. There were drug addicts, there were drug dealers, even up early in the morning. And then there were police officers, which is what I kind of wanted to talk to you about today, since that issue is so prevalent over the last few days. I'm just going to be as frank as I can about the subject because I think it's important from a scientific perspective, and I'm going to talk about that a bit too. So when I was in high school, I was routinely harassed by police officers regularly. What they would normally do if my friends and I were together, and sometimes even on the way to school in the morning, would be they would pull up a bunch of cars when they saw us, and they would all get out with their guns drawn, and they would make us all lie on the ground with our hands behind our backs, and they would go through our pockets routinely. Sometimes they leave us down there. I don't think it ever was over an hour, but sometimes it seemed like it was close to an hour. And this, again, just became a matter of routine as I was growing up. I had friends when that happened who said that once they left us that their money was gone or that things they had of value was gone. I never had anything, but then I never had any money back then either. So, beaten by police officers. At one time they did that. So sometimes that happened on the way to school in the morning. We were just trying to get to school in the morning. And we would be harassed. We began looking at the drug dealers as our allies, to be quite frank, because they were bullies, but they weren't drawing their guns and making us get on the ground and going through our pockets like the police were. I was beaten. One time that happened, and they arrested my friend, a friend who had a very close friend of mine. After his arrest, all of the charges were dismissed immediately because there was no evidence. But as they were arresting him, I didn't think it was right, so I got up and said I didn't think you should be arresting him, and that was followed by a series of clubs coming down on me. I laugh about it now. It's a very painful memory for me, and it was not a good experience for me at all. And at that time, my friends and I just felt that there was not a lot of justice for us. And as even recently, my best friend, who used to be with me those days, his son, 5 years ago, Raphael Briscoe, was killed by police officers in Southeast Washington in the daytime, shot in the back. And he's still suffering with that. Actually, I was with him yesterday. He's still suffering with that right now, trying to cope with the loss, and still trying to get some justice from that. Well, why is that relevant? Well, recent years, as these incidents have begun to be more increasingly broadcast, it's increased awareness, and that's a good thing. That does come with some pain for me because, now I have these conversations with a lot of my colleagues and a lot of friends that I've developed over the years, and they're like, the country really has a problem. The police officers are out of control. And then I say, well, the police officers have been out of control. But I'm grateful that now that it is becoming increasingly broadcast and people are becoming increasingly aware. And my dad is in news, he's a journalist. So I understand that a lot of times you have to, the news media and the journalists have to present an issue as if it's new, because that's how it gets the most attention. And as scientists, you all will be journalists also. Part of what we do as scientists is us being journalists. We have important questions we ask, we answer those in our lab, but at that point, we're really just beginning. And it's our responsibility to communicate those results and to try to see that those results help somebody. And that's what journalists are trying to do. But it's kind of bittersweet for me, for somebody who lived through those experiences, who went through those when I was in high school, who continued to go through those when I was in college, to hear so many people saying this is getting out of control or it's a new issue. But I'm here to tell you that it's not a new issue. It's been going on, and what's new are that a bunch of really bright, smart people, most of them located in California, a lot of them employed by Steve Jobs, decided they were going to change technology. And not only did they do that, they were able to make that technology popular, get it into the pockets of everyday citizens. None of us, probably them, had no idea how this technology would help or hurt our society. And that technology is now revealing a lot of the experiences that I went through when I was a teenager and a young man. And that is important to me because it's what I do for a living, and it's what many of you will do for a living. And it's important for me to say that when you're in your lab day in and day out, when you're running experiments, sometimes it's difficult for you to see the end of that. Will I really help anyone? will I really have an impact? As I'm sure some of those people working in the labs in Silicon Valley saying, how can we make cameras really small and put them on everybody, probably did not recognize, maybe some, but most probably did not recognize what would be the outcome of that technology. And that's why what we're doing is so important. It's very important. I don't want you to forget it. I love to do it, and I'm sure many people in the audience really enjoy doing it. I enjoy coming to the lab. I enjoy what I do. But it's important to recognize that what you do is important and has relevance. And if you do it well, it will have a positive impact. And I think my experiences when I was at Dunbar kind of reflect that. I'm not an engineer. You know, I don't build cameras. I'm a neuroscientist. But seeing this story unravel in the media to the extent it has over the last couple of days has helped remind me that what I'm doing in my lab is very important. And hopefully someday, some of the discoveries that we're making right now will have an impact that will make things better for people. And I want to leave you with that slide, that you know, that ethic can build tenacity and build resilience, and those are two things you're going to need to get through what you're doing right now. After I graduated from Dunbar, I went to Howard University. I like to joke that every school that I went to was geographically closer to my house. So I grew up right in Howard's backyard. That's Founders Library, which is on the main campus of Howard University. And I went to Howard still wanting to be a scientist. And I was fortunate enough to get into, actually I started doing music at Howard. That was in the late 80s, early 90s. And I don't know if you guys know, that's when hip hop was really burgeoning. And I was a part of that movement back then. So I can remember going to class and getting off of class and going to work and getting off of work and going to DJ parties. And if I would tell no one that as a sleep scientist, I have to advocate against that. I would not advise anyone to do that. But I was a sleep scientist back then. I did not know any better. I remember back then, my dad, I thought I was at a crossroads. I was like, Dad, I think I'm going to follow this music thing. And he was like, well, what do you want to do if all obstacles were eliminated and all options available to you? What would you like to do? And I was like, you know, I would like to be a scientist. And he was like, do that. A lot of the people that I was working with back then have gone on to become multimillionaires, so I sometimes think he regrets that advice. But back then, and quite honestly, back then, if I'd have known what that would become today, I might have thought twice. But in any case, I also like to tell the flip side of that was that I only know a few of the people we worked with that ended up being successful. The vast majority, in entertainment or athletics, you kind of roll the dice. The vast majority of people I worked with did not. And I'm one of the few lucky ones that had an alternative, which they call it an alternative. I call it my primary goal, which was to become a scientist. In any case, I got into a lab at Howard University. We were studying fireflies. So I got into a lab that had no money. So I would never advise that. I'm going to say, first of all, if you can avoid it, if you can't avoid it, try to go into a lab that is funded. Try not to go into a lab that has absolutely no funding. But I went into a lab that had no funding, and I know people that have gone into labs that have that had no funding and were still successful. So obviously there are exceptions to the rule. So we were looking at fireflies. We were interested in their vision. Fireflies had really have adapted a visual system that not only do they have a photopigment that absorbs light that allows them to see. They have photopigments that screen out certain wavelengths of light that allow them to focus in on the wavelength that they're flashing so they can mate. And at that time, the lab was really interested in identifying that photopigment. And my advisor had been going on years and years and years. He'd never been able to identify the photopigment. So when I got into the lab, my second or third day, it was probably my second or third week, I was dissecting these ommatidia. And I gathered a bunch of them, and I pulled out what looked weird to me, and I collected it, and I showed it to him. And he almost had a heart attack. It was the pigment he had been looking for. He looked under the microscope, he couldn't believe it. At that time, he thought I was a genius. He's like, this is the greatest thing walking. He's the greatest God's gift to the earth. So after that, he just left me on my own. He never mentored or advised me. He just said, you keep doing that. You collect as much of that as you can so we can run spectral sensitivity curves. Turns out I'm not a genius, I'm just colorblind. Anyone here with red-green colorblindness? Well, if you have red-green colorblindness, you understand, so there are shades of green that I cannot see, there's shades of red that I cannot see. Most, if not all, other colors look different to me than they look to most of you. So it wasn't that I was a genius, I'm just the only person that could see the pigment on the background of the ommatidia. So I was able to pull it out. whereas anyone wasn't. So, but that story has some negatives with it because he left me alone for 2 years. Every semester he gave me an A for independent research, and then when it was time for me to graduate, I gave him what I had collected, and he was not happy. It was not very clean. It had a lot of choroid in it. It wasn't collected in a fashion where he could run spectral sensitivity as is. So at that point, he gave me an F. My very last semester gave me an F for independent research, which was going to prevent me from graduating. But I wasn't having that. I went to the dean. The dean found out what had happened, said, you're being railroaded, and the dean allowed me to graduate. And they got him to change the grade. That's important for me. I had a falling out with him at this time, but that was important for me because I learned a valuable lesson from that. And quite frankly, so his goal was to keep me in the lab for another year. You take this F, you stay in because you can't graduate with an F in independent research, you stay in the lab for another year, get this right. And had I known now what I knew then, I would have stayed. A lot of my colleagues that know that story say, you can't allow yourself to do that. But I would have. It would have gotten me one or maybe a series of publications as an undergraduate, which would have been much more beneficial to my career than graduating a year early. And it would have allowed me to repair my relationship with my mentor, which is very, very important in the long run. I do recognize that what he did wasn't necessarily right or wasn't, I don't think he treated me very well, but I also recognize that I allowed him to just let me be independent for 2 years, and I should not have done that. And one of the things I want to impress that I learned from that experience is that a mentor-mentee relationship is a two-way street. It's not as a teacher-student where you come to class, you take notes, you take tests, you pass the class, teacher gives you a grade, and you move on. The relationship with your mentor, you have an advisor, and the best case scenario is that your advisor becomes your mentor, but they're two different things. And once you have established a relationship with the mentor, you have as much responsibility as that mentor for your training and for your productivity. And that's what I learned from that experience. It takes an ability to humble yourself. It takes an extraordinary amount of humility. But when you understand what your goal is, then it's not hard to do. So, as Michelle noted, after I left Howard, I was DJing, I was doing music, and I moved to Atlanta University, where I attended Georgia State University. So initially, at the time that I graduated, Atlanta was the burgeoning scene for hip-hop music, so I decided, I would go down there and draw my hat, see what I could do about it. And I always wanted to go to grad school. Even when I graduated, I am going to Johns Hopkins. I'm going to study neurobiology. The lab I was in at Howard University was a neuro lab. So I said, I'm going, but hip-hop was kind of really taking off. A lot of my friends had gotten recording contracts and gotten these big sign-on bonuses, which gave them a lot of money, but that turned out not to be so good. In any case, I said I'm going to go to Atlanta because the music scene down there seemed to be more independent. And Michelle is absolutely correct. In addition, I had a daytime job, and in addition to being a music producer, I got a gig at 88.5 WRAS Saturday nights DJing their hip hop show, and the radio station just happened to be on the campus of Georgia State University. So it's true. I said I'm going to grad school anyway. I might as well walk over to the neurobiology department and see what's going on. And there I met Elliot Albers, a great guy. I told him about my experiences in grad school. He had been so happy because I'd worked on mammalian and frog retinas. My experiences at Howard. I'd worked on mammalian and frog retina, and he was really excited to get somebody that worked on a retina because a burgeoning story in circadian rhythms at that point was trying to find the circadian visual system. What is the system that allows your circadian timing system to see light? Because we had known at that point it wasn't through visual photoreceptors. So he asked me to join the lab as a master's student, and I was like, okay, I'll join as a master's because I'm going to Hopkins. So I joined as a master's student, and then about a semester after I joined, a group in University of Virginia discovered the visual system that trains circadian rhythms. So I had mounted maybe 100 retinas for Elliot at that point, and got completely scooped by the group at University of Virginia that found a new photopigment melanopsin, and the retinal ganglion cells sends the signal to the circadian timing system. So the system that entrains your daily rhythms through your eyes is different from the system that you're seeing me and seeing these slides right now. They're completely separate systems. And that alternative visual system, the melanopsin system, was discovered by the circadian group at the University of Virginia, who I went on to work with after that. But Elliot, to his credit, convinced me not only to stay at Georgia State, but to enroll as a PhD at Georgia State University. And quite frankly, I think it was the best professional decision I ever made in my life. And the reason it was such a good decision, and the reason it continues to pay off in dividends for me, is because Elliot and I had developed a really sincere relationship as a mentor and a mentee. He was willing to teach me everything I needed to know to become a productive scientist. And if you have that, take advantage of it. Value it. That is, I always like to say, if someone's done something before you, it's better if they just tell you how to do it instead of you learning the hard way, right? So Elliot had an immaculate training record, and he was a successful scientist, and he had taken a liking to me. And he said, if you stay here, I guarantee you'll have publications, you'll get cited, and we'll get you into a very competitive postdoc. And I believed him, and he did exactly that. So during my time with Elliot, I was able, I think, through the lessons I learned when I was an undergraduate, kind of turned the tide. I was able to humble myself and to be taught, to sit in a position where I could be taught. And I try to do that for my students in my lab right now. I try to create a space for students to make mistakes. I believe in mistakes. The rule in my lab is you can make mistakes just as long as they aren't the same mistake. Make new mistakes, because if you're making new mistakes, that means you're doing something new. And if you want to learn, you're going to make mistakes. And Elliot had that patience with me, and he allowed me to do that. So we studied, I moved on from the visual system in the retina, and we moved on to look at the hypothalamus, and my PhD thesis, which, not to brag, but yielded a series of papers, looked at how light information was transduced from the optic nerve into the hypothalamus to entrain circadian rhythms. So I guess the take-home message from my experiences at Georgia State University is if you find a mentor, again, that is a two-way street. You have the responsibility to try to work with that, to not only be taught, allow yourself to be taught, but if you're lucky, you may even teach your mentor things also. And I always like to say some of the greatest people I've ever met and some of the people I've learned the most from are students that have been in my lab. So after I did my PhD at Georgia State University, I went on and did a postdoc at Northwestern University. This is a picture of Northwestern. This is a lake that's on the campus. That's a much smaller lake. On the other side of that building is Lake Michigan, which is beautiful in the summer, not so much in the winter. But I had a lab and an office that overlooked Lake Michigan. And from there, I moved into the field of sleep, which I'm still in that field right now. And I learned to transition from generally looking at daily circadian diurnal rhythms to looking at one specific circadian diurnal rhythm, and that's the sleep-wake cycle. And during my postdoc was where I got into I'm a neurobiologist by training, but we were really interested in the genetics and molecular biology of sleep, which is what I study now. And I was in the lab of Dr. Fred Turek, where we began those series of studies. And I also began looking at sex differences and sleep mechanisms and sleep-wake disorders in that lab. But one interesting thing for me to be at Northwestern, and it may resonate with some of you, was before that I was at Dunbar High School, I was at Howard University, and even Georgia State. But when I went to Northwestern, I had a unique experience where nobody there could relate to my experiences at all. I had an opportunity to teach some courses when I was a postdoc, and even though those courses were neurobiology-based, anytime the subject of medicine, neuromedicine came up, and we were talking about the actual patients, it was a difficult experience for me. And I have an undergraduate in my lab now. He's an undergraduate at Swarthmore in Pennsylvania, and he's a Howard Hughes Medical Institute fellow. And he's in my lab. Howard Hughes gave him the opportunity to pick where he wanted to do research for a summer, and he came down to work with me. He's African American, and he's been talking about his experiences at Swarthmore and how, even though he's able to compete on an academic level, it's very difficult on a social level because a lot of his experiences are kind of discounted. And I hope that I'm able to help him work through that, because I went through that when I was at Northwestern University. And at times it'll make you question what you're doing. It'll say, well, yeah, I know that the science I'm doing is important and impactful. I know that I'm productive, and I know that I'm having an impact where I am now. But you sometimes worry if you'll have an impact on a larger society that really doesn't have the patience for your personal experiences. But that's only a perception. Fortunately, Northwestern University isn't reflective of the larger society, and I learned that once I left. So the experiences there taught me that I have to be able to relate my work as a scientist and my findings to a broad variety, a broad cross-section of people. If I want my work to have any sort of impact at all, I cannot just restrict it to the people who understand me as a person or the people who understand my social background. I have to be able to explain what I'm doing, report my findings, and report their importance and significance to even the students at Northwestern University, even though they may see me and look at me sometimes and say, why is this guy standing here? And that was an experience there also. So scientifically, everything, I didn't really have very many challenges at Northwestern. The lab was well funded. The working group I was working with was very supportive. But I'd have experience in classes when I'd be talking and subjects of medicine would come up and we talk about health disparities. And people would say things like, well, if you don't have any health insurance, you're lazy. I just used to hear that from students all the time. Or, if you come from a disadvantaged background and your parents are lazy. And some of those things were difficult. And I don't know, there may be a few people in the room right now that go through those experiences. And I'm here to tell you, there's light at the end of the tunnel. Those experiences make you stronger. They help give you an insight, again, to a broader mindset. And in the long run, they help improve the science that you're doing at the bench. And then from there, I went to Morehouse School of Medicine, where I still am physically, but I'm in the process of moving. I've been in Morehouse School of Medicine for 10 years. I was recruited to Morehouse from the director of the Neuroscience Institute and the chair of the Department of Neurobiology, Dr. Peter McLeish, who was revered at the Neuroscience Institute at Morehouse School of Medicine. And I set my lab up, and we've watched our program grow. When I was recruited there, there was one circadian biologist, Gianluca Tassini, who also helped recruit me there. Since my recruitment, we've recruited several others, Alec Davidson, Jason DeBrine, Chris Allen, and we've started a circadian rhythms and sleep disorders program, and the school is now trying to create that into a center. I have a pretty strong working group, most of us supported by NINDS. And we've, I think, had some pretty impactful findings since I've been there. So I like to say that even though I'm leaving there, the circadian and sleep piece is I think, stronger than when I left it. So what did I learn since I've been in Morehouse for 10 years? Morehouse is a small school, small, historically black college and university in downtown Atlanta. And the med school, even though it's affiliated with Morehouse College, is independent. So we're a small black school, small underrepresented school. Don't have a lot of resources, so I've had to learn how to navigate. I came from Northwestern University, which is kind of the other end of the spectrum, and I was drawn to Morehouse just by the quality of science I could do there. I knew that Peter was a great mentor, but what I learned there most is how to communicate. Byron Ford was also a mentor to me when I got there, and he's now at University of California, Riverside, and he gave me some advice when I first came, because when I first got there, it's a small school. Sometimes it's difficult to get things done, right? But he said this is an opportunity, because what you have to do is you have to be able to communicate your science to everybody here. When you're going to sponsor research and you have a grant due, you have to be able to communicate your science, to tell them what it is you're doing, get them excited about what you're doing. When you're talking to the cleaning staff who hasn't been emptying your waste baskets, if you can get them excited about the science you're doing, they will make extra trips to your lab to clean your waste baskets, to the animal care staff, to the administration, to the president of the university, to the dean of the university. You get yourself into the habit when you meet these people. Don't just talk about the weather or talk to them about what you're doing, not because you're selfish and talking about yourself, but talk about your science and what it is that excites you about your science. Learn how to communicate what you're doing to a broad cross-section of different people. And that's what I learned when I was in Morehouse School of Medicine, how to do that effectively and efficiently. And pretty soon you're going to have people that are not scientists, people that have no idea what the fundamental things you learned are, helping you. saying what this guy is doing is exciting, what this young lady is doing is exciting. I'm going to do whatever. If they need something, if they have a grant and it has to get in and they came to me at the last minute, I'm not going to complain. I'm just going to get it in for them because I understand what they're doing is important. If they have an invoice that hasn't been paid because somebody in the lab didn't submit it correctly, I'm not going to berate them. I'm not going to get on that person. I'm just going to get it done because I understand what this person is doing is important. And that is something that if you learn it, the sooner you learn it, the more beneficial it will be to you. And this is where I am relocating, the UCLA, University of California, Los Angeles. I am already a faculty member there, and I'll be there physically in the next month or so. So with that, I'd like to go into a few suggestions for becoming a successful scientist. I am not going to be able to get through all of these slides. I'll get through as many as I can, and then I'll stop and open the floor up for questions. So I talked about this, a supervisor versus a mentor, an advisor versus a mentor. A mentor relationship is one that you have to nurture, that you have to fertilize, that you have to help grow, that you have to keep monitoring. And I strongly recommend this. You can avoid a lot of hassle if you have somebody who's an expert teaching you what you need to do to be successful. Get involved and take responsibility for your own matriculations, for your own research, something I did not do as an undergraduate. I had to learn that the hard way. Work hard. I don't have to say that too many times because if you guys weren't working hard, you wouldn't be here. Play hard. When you're young, I have a family, 3 kids, a wife. That really it changes your life. So, and some of you may have a wife and kids and you can relate. I always tell young people, have fun. Have fun. Take advantage of opportunities to travel. Take advantage of opportunities to come places like this and meet people. As Michelle mentioned earlier, she and I met at events like this. And have fun. Learn together and play together. Be creative. Be proactive. Write. Work on your writing. Work on your writing. The standard I hold myself to is the standard of another mentor of mine. His name is Gary Gibbons. He's a director of Heart, Lung, and Blood here. He was at Morehouse with me, and I worked with him for years. And he managed to get everything he wrote either published or in a grant. I mean, everything. If he sends an email to you, something that what he wrote in the email is going to end up in the paper or end up in a grant. I was floored by that, and that's the standard I hold myself to. Everything I write, even if it's a note I write to myself, some kind of way, this has to be published. Read literature, go to seminars. You guys are here, so I don't need to really emphasize that too much. Do presentations as much as possible. I sat on the dissertation committee for a very bright student. We had a meeting last week, and she was going. Her presentation wasn't polished, and her advisor kept giving her advice, and I had to tell the committee, no, what she needs is to go to meetings and give presentations. That's where you're going to become the most polished, is when you're actually presenting in front of your peers and your colleagues and people that are going to be honest with you. And plan for your future very, very early. And with that, I think I will stop and open the floor up to questions. Thank you. Yes.
Hi, thank you for talking with us today. I currently work with elementary schoolers from West Baltimore and inner city Baltimore, and I know that they're at risk to not being in school, and I was wondering if there's any way I can better reach them and help them find kind of a core or a seed in academia to help keep them going.
Yeah, that's a tough one. And actually, Morgan State University, which is in Baltimore, has a program that I think it was in the Washington Post yesterday, that's very successful, with convincing people who had dropped out of school to return, because they recognize that these students are kind of a valuable cohort because they've had experiences in academia, and since they're dropping out, they've gained additional experience. They've learned how dropping out has affected them, whether for better or worse, and they're recruiting them there also. For me, I'll be quite frank. In the environment I was in high school, there were a lot of negative influences from my teachers and counselors, people that were supposed to be helping me. There were positive influences also. If I could go back, I would have wished that those positive influences could drown out the noise of the negative influences. So that would be my biggest advice, is to find out if there are people in authority that are kind of motivating that kind of negative outlook, and seeing if there's a way you can combat that. And also just reminding kids, especially kids that are most at risk, that don't get that many positive or reinforcing influences, that the people that are negatively influencing don't know them, don't know who they are. And the people who I had in my life then who were positive influences had to continue to remind me that, you can't let other people, even if they're an authority or a teacher, tell you you cannot be a scientist. If you know that you want to do that and you know you have the tools, the ability and the aptitude to do that, then you can do it, or whichever way. And just to remind them that, don't trust anyone's opinion about yourself over your own opinion about yourself. And for me, I think that being reinforced from elementary school all the way up to now was really a motivating influence for me. It kind of, made me a little cocky, but I'll take it. Where it's just like Ketema, you have the ability to do it. You don't have to listen to teachers that tell you that you can't do it.
Anybody else?
Thank you.